Dr. Robert Brown, the Chair of Neurology of Research and the Chair of Department of Neurology at UMass Medical School. He is an internationally known researcher and physician leading us in our quest to find a cure for ALS.
Well, thank you very, very much, Abby. It is a privilege to be here, and above all, to take just a minute or two to tell you where things are going in terms of some of the ALS research. And in addition, to take advantage of this moment to express on behalf of all of us who are doing ALS research here our very deep thanks and gratitude for what you're doing. I've been trying to think about ALS and involved in ALS research, if you can believe it, since 1979. And over the years, one finds that the resources that are applied to pay the bills are, of course, enormously important, but equally or more important is you see the word inspiration, it's motivation. And of course, as one goes through decades of this, there are legendary motivators. One thinks of the name Delvecchio, Kennedy, Mazareas, and now, of course, the Frates family. What you have done, apart from the phenomenal fundraising to motivate and inspire, is almost beyond words. We're profoundly grateful to you. So without a lot of time left, I thought I would just walk you through some of the things that we think are particularly important. I'm going to tell you a little bit about one particular program for which your support has been absolutely pivotal and will be. It's a therapeutic avenue that hopefully will take us to a human trial within a year and a half. And then I'd like to tell you a little bit more about some of the other things going on here at UMass. So I'm going to say first a few words about where we are in terms of genes and gene silencing. This cartoon before you is kind of an obvious portrayal of how some of us think about ALS. There are some cases that are clearly genetically endowed, and there are others that don't run in families, and we don't know what causes those except perhaps environmental factors. And the question is, how do you get from genes and environmental factors all the way to ALS? And so our view, and it has been our view for about 30 years, is that if you study carefully what these genes are and how they misbehave to cause ALS, you'll define a pathway. And that pathway, in turn, defines opportunities for treatment. So this is taking bad news and trying to turn it into good news. And the treatment could be small molecules, it could be drugs, it might be approaches to essentially blunt inflammation in the spinal cord, it might be cell therapy, and indeed we're engaged in stem cell therapy now jointly with the Mass General, or it might be other techniques such as trying to go way upstream and treat the offending genes before they can cause too much trouble. How does that work? Well, let me talk about that because I think this is a very important strategy. When we last convened, I think I dug it up this afternoon, a PowerPoint that said we had 25 ALS genes. Well, there are now close to 45, and there are a couple others in press. So the landscape of ALS genes that misbehave and can trigger these death pathways is continuing to expand around us. A whole nother topic, very interesting. How do you lump these different possible causes together to get the big ideas and not lose yourself in the weeds of all of the small details? But in fact, most of these genes, in one way or another, do something that's toxic, that's poisonous. Here, for example, is the first ALS gene, SOD1. Normally, it's an antioxidant that helps keep things alive. So normally, it's very important to keep a motor neuron healthy and muscle healthy, and that's what I've tried to show here. But when it's mutated in ALS, in fact, it makes a poison which kills the motor nerve. It is an adverse active killing process, which of course then causes the disease. And we know that, of course, this is a phenomenon we see in people. We also know, as you know, perhaps some of you saw this upstairs, that if we take the gene that causes the same disease in people and put it into animals, we can generate an animal model which is to an eerie degree a lot like what we see in our patients. The upside, of course, is that we can do things with the animal models to look for treatments that are not possible in people. But the point is this, and this is the take-home, that if many of these genes act by doing something that's poisonous, if we can turn them off and prevent them from creating a toxic product, we'll have a therapeutic. So that's the strategy that we've been pursuing very aggressively here at the University of Massachusetts. I think I feel very blessed, in addition to having this extraordinary ALS Angel Fund family to work with, to in fact have an incredible team of scientists here who are pitching in and working aggressively on this. So we're lucky to have one of the country's few human gene therapy trials unit headed by the fellow at the left, Guangping Gao. And then we have 3 extraordinary scientists who are using gene therapy to help get into the nervous system to turn off genes. How does that work? Well, the nervous system has an iron curtain around it, and it very well should, because you don't want things getting willy-nilly into the brain and the spinal cord. There's a blood-brain barrier that's meant to keep things out. And so part of the challenge is how do you get, if you've got a treatment, how do you get it in where you need it to work? And one of the things that's become clear is that you can use viruses to carry the treatment through the iron curtain, through the blood-brain barrier, where they will finally work within the nervous system. And so these extraordinary investigators have found several ways to take viruses, put in them the things you want to treat the nerve cells, and get that into the brain for therapy. Now, what's shown here is one amazing, one of several rather extraordinary results that has come from Dr. Miguel Sena Esteves and his graduate student, in which he's taken those ALS mice that you just saw, and he's treated them, or not treated them, and said, how long do they live without or with treatment? And the black line here shows you the survival curve for the animals that do not get treated. Basically, they're all dead by about 150 days, and it's a look-alike dead ringer for ALS. It is ALS. But if he treats them, what you can see is that there's at least 1 month to 2 months prolongation of survival. And while that's not a complete cure, that is a huge impact on this particular model. So we're taking this very seriously and asking, can we think about getting this into people, at least for those people who have this kind of ALS? So we've been also privileged to work closely with Dr. Chris Mueller, who's in the audience, to say, can we now try to turn off this offending gene in not a mouse, not a petri dish, but in a mammal, in a monkey. And so we've embarked on this aggressively, and one data point is shown at the top right there, and that is that if you use the virus to get the silencing agent into the monkey, in fact, you can suppress activity of this, in the best experiments, of this gene almost completely. So our hope is that this might set the stage for moving into a human trial, and it's this that we're embarking on with keen passion and great interest. So I won't go through all of this flow chart, but basically we've completed a number of preclinical studies, and what we're now doing is working through some additional monkey tests so that we can go to the FDA and we can say we've done this with the right dose, with the right virus, with the right purity, and we know it's safe. Now let us do it in people. And our goal is to try to do it in 10 to 15 people. We're going to do this jointly with Merit Sokovich at the MGH. The extraordinary work that I've just mentioned here will be the basis for this. And we hope that ideally, by the end of next year, we will be into the human trial. And this will be, I think, if this doesn't treat all of ALS, at least it's a beachhead in treating one of the most aggressive forms of the disease, which is ALS that arises because of SOD1 gene mutations. Well, just to take another 4 or 5 minutes of your time, and then I'll stop, I want to mention 2 or 3 other kinds of things that are going on here and why we're so profoundly grateful for your continuing support. One of the reasons I left the MGH after 30 wonderful years there is that there's a group here called the RNA Therapeutics Institute, which is specifically built to think about how we can get these kinds of ideas into therapies. And the people who run it, just briefly on the left, is a fellow, Victor Ambros, who discovered one of the major gene silencing tools. He got the Lasker Award for that last year. Craig Mello, who got the Nobel Prize for his discoveries of how to turn off genes. And you put those together with people like the gene therapists, and you have a real powerful group. And there are many affiliated faculty who then take the ideas from the directors and from elsewhere and try to implement them into investigations that will lead to treatment. One of the individuals whom I hold in the highest regard is Dr. Fenbiao Gao, who's here, a man who came to us with a lot of expertise in thinking about the overlap between ALS and frontotemporal dementia. Believe it or not, there are some people who unfortunately become demented as they get ALS because there's shrinkage, not just of the spinal cord, but of the whole brain, and I've tried to show that in this picture here. So Dr. Gao has several ideas about how that occurs, and he has models both in mice and in flies of why it occurs. He's also been an expert in studying development of the brain. Dr. Larry Hayward, who's here, has worked with us, with Daryl Bosco, to look at another gene called FUS, which when mutant causes terrible forms of ALS. We've now seen an 11-year-old girl, a 14-year-old girl, an 18-year-old who have mutations in FUS who survive less than a year. This is a house of fire. And so if we can understand how this gene works, it too will be another beachhead. This is unthinkably terrible. I will just point out that this gene was discovered by one of the Angel Fund Fellows, Tom Kwiatkowski, when he was an Angel Fund Fellow back with us at the Mass General. So Larry and Daryl are doing fantastic work. They built a mouse model, and now they're studying how it works in a Petri dish. I don't know if it'll work, but in the bottom you'll see a bunch of cells with green in them. That green is the sick protein, and what we're doing is stressing the cells a little bit here, and you'll see that the sick protein forms clumps, which are called stress granules. And what these do, in effect, is pull some of the most important parts of the cell into little isolates where they're no longer available for normal function. And this is something that these guys can do in a Petri dish. It's part of the disease, and it gives them a very powerful way to think about developing new therapeutics. I want to mention also the work of Mark Freeman here. One of the extraordinary things in biology that people have known for probably now 150 years is if you cut a nerve, within about a week it degenerates. It just essentially completely falls apart. And what Mark Freeman here, who's a Howard Hughes investigator, has learned is that when it falls apart, it's not just because it's not getting what it needs to survive. It falls apart because built into every nerve, get this, is a suicide pathway. And when the nerve is injured, for some reason in evolution, it's been useful to say it's now going to destroy itself and clean up and go away. And what Mark has done is discover a series of molecules that cause that process to occur. And is now looking at ways we can inhibit that and asking, does that have a bearing on a disease like ALS? He's also made a fruit fly model of one of the other ALS genes, TDP43. We're also thrilled to have a young investigator, Dr. Nils Henninger, who has said, can I take this information about nerve suicide and use it to understand other very basic processes, one of which is what happens in head trauma. When you injure the head, does that somehow activate the suicide pathway in a way which is adverse? And if you inhibit that, will it slow the effects of head trauma? And while head trauma is by itself hugely important, it's also relevant to ALS because, as we've all read again and again in the newspaper, there are these terrible stories of athletes who suffer head injury repetitively, particularly football players, who go on to develop brain degeneration with this so-called encephalopathy. And one of the things we would like to do is see if we can use this new insight into something like the suicide cascade to understand how that terrible form of ALS and dementia can occur. Last minute or two, Dr. John Landers, who is another Angel Fund Fellow, has worked with me and with some wonderful investigators in Israel where we found a new gene, family in southern Israel, that's important in determining how rapidly a nerve can grow and grow out. And we've now, a year and a half later, got a mouse model of this. One was graded here, another with a colleague at Arkansas. This is a mouse that has the Israeli gene in it. It's a mouse that should be laughing, jumping, standing around, climbing the walls, and in fact, it can barely crawl. And this is a consequence of having mutations in this Israeli profilin gene. Why is this important? One of the things we want to do when we have a test drug is say, does it treat the SOD mice? That's fine. But what if it treats all the other models of ALS? Then we've got something that's uniformly more applicable. And until recently, there have not been enough good mouse models of ALS. We think this will be a very important second model. And I'll just go on to conclude by saying that the most commonly encountered mutation in the ALS gene is in the funky name, a gene called C9orf72. And over the last 2 years, a remarkable group of young investigators whom I've tallied here, all of them, I won't read their names, but these are incredibly hardworking, either doctoral students or technicians or postdocs who have worked with us to generate a new mouse model of this new gene. And it has a number of funky features, including deposits of protein and RNA where there should be none, as shown in the bottom left figure. So this is, I think, another very powerful tool, because one of the things we're trying to do with this, as we're doing with SOD, is turn this gene off to see if we can prevent it from misbehaving. Last moment, I want to say that we and others in 1996 formed a group called the Northeast ALS Consortium. We've met frequently to look for ALS therapies. We've enrolled more than 4,000 patients in ALS trials. We now have 110 centers, not just in North America, but also in Europe. And through that group, there are a number of approaches to trials that we can talk about later, but at least there is much in the pipeline to occasion hope. And finally, this is our group at the Timlin race this year, all of whom join me robustly in saying thank you to you.